Hi everyone, my name is Princess G Magdamit and I will be your medtech for today. And now, I will be showing you how to do the paper boat making for the histopathology laboratory. Let's go! First, we must fold the index card vertically and horizontally. Align the folded vertical and horizontal lines. Repeat this step on the other side of the index card as well. A formation of cross or X-like folded lines will then appear. And after that, divide the paper in 5 equal columns. Also, divide the card or the paper in 3 rows equally. The next step is known as the sandwiching part, in which we must tuck in the sides along with the lines backwards, just like what is shown in the video. Repeat this step on the other side as well. And we're done! Hello everyone, it's me again, Princess, and I will be teaching you on how to do the specimen receiving. Yes, so today, I will show you on how to handle when receiving histopath specimen. As we receive the specimen, we must always be careful and make sure that the important information is written on the container. Now, we must get the logbook to record the arrival of the specimen and also write down the important details. And lastly, we must ask for the name and signature of the person who brought the specimen to the laboratory will also be written in the logbook. And that is how we receive specimens in Histopath Lab. Thanks for watching!
Hello everyone, I'm CJ Presto from BSMLS 2C, and I'll be the one who will present to you the accessioning portion in histopathology. Since we're inside the lab, it is important to wear necessary PPEs, but in our case, since we're just going to be performing accessioning, we can opt wearing surgical gloves, but just make sure that you still disinfect your hands. So let's start! After receiving and filling up all the necessary documents, we can now proceed to specimen accessioning. In this section, necessary documents such as request form, patient description information, site of origin and history, patient's receipt of transaction, and others are needed. Moreover, we also need to fill up the patient's serial number by following this proper format. Type of specimen, dash, the last 2 digits of the current year, dash, specimen control number. Note that we have 2 types of specimen, based from where we acquired it. It can either be AP, or known as autopsy pathology, and the second one will be SP, or surgical pathology. In case-to-case basis, if ever we will be receiving multiple samples in one patient, we will be designating specific letter after the control number. Note that each letter will serve as the specific sample from that patient. After assigning the patient's serial number, we can now write all the details in our logbook. Such details are the serial number, patient's name, age, gender, room number, specimen, name of the pathologist who will examine the sample, date of specimen received, and of course, the due date. After writing all the pertinent details, we can now proceed to filling up the surgical pathology worksheet. In here, we will be writing all the necessary annotations during gross examination and the microscopic description of the slides done by the pathologist. Remember, everything must be written. Nothing more, nothing less. And last but not least, after performing the worksheet, all the documents that we have gathered during the accessioning process must be attached in one folder. Remember that specimen rejection is possible if the criteria are not met. These are unlabeled specimen or wrong labeling of the specimen, incomplete patient's information, if the specimen is left unfixed or unrefrigerated for a period of time, putrefied or autolyzed specimen, damaged specimen, insufficient sample for processing, spilled or contaminated sample, failure of the requesting physician to enter the request in the computer, empty containers without the specimen or the form itself, and others. So that's it, everyone. I hope you enjoy my demonstration. Thank you and God bless.
Hello everyone, my name is Tara Sanyal and I am here to demonstrate to you on how to prepare fresh tissue. The methods of tissue examination may vary according to the structural and chemical components of the cell to be studied. Depending on the situation, fresh or preserved tissues may be examined. Fresh tissues have the benefit of being examined while still functional, allowing protoplasmic functions including motility, mitosis, phagocytosis, and pinocytosis to be observed. However, because tissues examined in their fresh form are not permanent, they are prone to develop the alterations that are commonly found after death. So, the methods of fresh tissue examination includes teasing or dissociation, squash preparation, and smear preparation. Smear preparation have 4 types: the streaking, spreading, pull apart, and touch preparation or impression smear. But first, from the frozen block of tissue, cut several small thin slices of tissue. Immerse the tissue slices in a petri dish filled with saline solution. Using a dissecting needle, carefully tease the tissue slices until they unfold and spread out. Carefully mount a teased slice on a slide and cover it with a cover slip. And label the slide. Add 1 to 2 drops of methylene blue stain directly on the tissue slice and the glass slide. Cover the stained tissue with a cover slip or another glass slide. Apply enough pressure to crush the tissue flat and label the slide. Pour a small amount of the sediments on one end of a clean glass slide. Using an applicator stick, streak the sediments in a straight or zigzag line throughout the remaining length of the slide. Cover it with a cover slip. Label the slide. Transfer a small amount of the sediments on one end of a clean glass slide. Using an applicator stick, spread the sediments throughout the slide. Cover it with a cover slip. Label the slide. Place a drop of sediments at one end of a glass slide. Cover is with the end of another glass slide. A single uninterrupted motion, pull the 2 slides apart in opposite direction. Cover the smear with a cover slip. Label the slides. Cut the black in half, exposing a fresher surface. The freshly cut surface to come in contact with the surface of a clean glass slide. Apply gentle pressure, allowing the cells to be transferred directly to the slide. Cover the smear with a cover slip. Label the slide.
Hello and good day. I am John Michael Sardan and I'll be your medtech for today who will demonstrate fixation. So what is fixation? Fixation is a histological technique that aims to preserve the sample's cells and tissue constituents with the least alteration from its living state with the use of fixatives. These fixatives are chemicals which disables the breakdowns of cellular components via autolysis, and it also disables its lysosomal activities. So before we get into the actual process of doing fixation, we must also make sure that we will be safe upon doing this process. How do we do that? By wearing our complete PPE. As you can see, I'm already wearing my complete PPE with my gloves, my lab gown, and my mask to prevent any accident to happen due to spillage of chemicals or accidents of cutting. Before we begin fixation, we must first characterize the specimen at hand. This process is called gross examination. In gross examination, we will be pointing out the characteristics as well as the dimensions of the specimen. So let's start. As you can see, the specimen has a bright red color in the outside, but in the inside, it is somehow pinkish brown. Then, its texture is smooth and soft. So after that, we shall now take its dimension. So start off, the length of the specimen is around 2.5 centimeters, while its width is around 1.75 centimeters. After performing gross examination, we can now begin with our process of fixation. So now, we will cut our sample into its respective representatives. So we will make 3 pairs, 2 cross sections and 1 longitudinal section. So with the use of a knife, cut your specimen around 2 centimeters squared, but not thicker than 4 millimeters. So there you have it. We have two pairs of cross-section and a pair of longitudinal section. Next, we will put our tissue representatives into tissue cassettes, or if you don't have one, you can use a substitute which is a gauze pad. And now for our last part, we wrap it up. After completely tying up your sample securely, we will now proceed and put them into our fixative. So we have here our fixative, which is 10% formalin, but we used a substitute of water since we don't have any formalin around. So we put it there and submerge these 3 samples for 24 hours so that the fixative can do its job. Note that every fixative has different times of its accomplishment of its job. So for 10% buffered formalin, it will take around 24 hours to fully fixate our specimen.
One of the steps of tissue processing is decalcification. This is the removal of calcium ions from a bone or calcified tissue through a histological process. Hi! I'm Ella Ixomo and let's decalcify together. In this procedure, we're gonna need our formalin-fixed specimen, our decalcifying agent, a clean tissue, forceps, and gauze. As soon as fixation is complete, take out the bone from the container and put it on the clean tissue. The fish bone is then placed in a gauze bag and suspended in liberal amounts of decalcifying solution by means of a thread to ensure complete decalcification and to protect the tissue from any precipitate that might be settled at the bottom of the container. The thread used in this method should be dipped in melted paraffin wax to avoid destruction of the thread by the corrosive action of the acid. Now our specimen is ready for decalcification. Tie a knot on the thread to refrain the specimen from sinking in the bottom of the container. Leave the specimen for over 12 hours or overnight for best results.
Hi, I am Michael Angelo A. Garcia, and I will be your method for today, and I will be the one to process the dehydration of the tissues. But first, before we start the processing of our tissues, let's wear first our PPEs. This includes the gloves, our lab coats, and our face mask. So the first procedure is to put the samples on the beaker with 70% ethanol and subdue it within 15 minutes. So the next procedure is to remove the sample from the 70% ethanol and place it to high percent of ethanol, specifically 90%, and subdue it within 15 minutes. So the third procedure is to remove the sample again from the 90% ethanol and place it to another higher percentage of ethanol, specifically 100%, and subdue it within 15 minutes. So the fourth procedure is that after 15 minutes, remove the sample and place it with another 100% of ethanol and subdue it with another 15 minutes. The fifth procedure is that after 15 minutes, remove the sample and place it with another 100% of ethanol and subdue it with another 30 minutes. On the sixth procedure, after 30 minutes, remove the sample and place it with another 100% of ethanol and subdue it with another 45 minutes. Lastly, wrap in foil and store your sample. The purpose of this dehydration is that because of the paraffin in which the tissues are embedded, it is not miscible with water and does not penetrate the tissue effectively. Hence, the water in the tissue should be removed before embedding. That's why this process is called as dehydration.
Hello everyone, I am Jazila Gail Puga, and for today, I will be demonstrating one of the routine procedure in histopathology laboratory, and that is clearing. Clearing or dealcoholization is the process whereby a dehydrating agent is removed from the tissue and replaced with substance that will dissolve the wax with which the tissue is to be impregnated. This step is done after dehydration and done before impregnation. So after finishing the procedure of the clearing, we will then expect to have a translucent appearance in our tissue sample. But before we start our demonstration, we should make sure that we are wearing our personal protective equipment, or PPE, for our safety. Today, I am wearing my lab gown, my gloves, and my mask. Sadly, I don't have my head cap, but I made sure to tie my hair up for my safety. And without further ado, let's start our clearing routine procedure. The first step of our clearing procedure is that from 100% ethanol of dehydration technique, we will immerse the tissue sample to our clearing agent, which is the water number 1, and leave for 20 minutes. And after 20 minutes, immerse again the tissue sample to the water number 2 and leave again for 20 minutes. Then after 20 minutes, immerse again for the last time the tissue sample to the water number 3 and then leave for another 45 minutes. And after 45 minutes, we will remove the tissue sample from the water and place them on paper towels to drain properly. And to finish the procedure, we will unwrap the tissue sample from the gauze pad. We will know that our clearing procedure is completed if our tissue sample appears translucent. And after that, we will wrap it in a foil and store it for the next histopathologic technique.